Hey everyone. Okay, so at this point we've covered photosynthesis. Basically, we looked at how photons from the sun provide energy to this important organelle in plants, the chloroplast, um, and how the chloroplast use that photon to create organic molecules such as, whoa, what am I doing here? Such as glucose. That's an ugly glucose, but let's call it glucose. Um, Also, a byproduct of photosynthesis from the light reactions, you might remember, is oxygen. And this is great because we will use both glucose and oxygen in our mitochondria to essentially produce the ever important ATP. So we're going to feed glucose, we're going to feed, or the byproducts of glucose, you'll see. And we're also going to feed it oxygen. And our mitochondria is going to take both of those ingredients or reactants, and we will use that to produce this ever important ATP, the thing that keeps us alive, as you will see as we go through the lecture. In the process of breaking down sugar with the use of oxygen, we're also going to produce water and CO2, which you might remember are important for feeding the chloroplast that's going to be doing photosynthesis. So they're going to use these as their reactants and give us these products, whereas we are going to use these as reactants to produce those products. So there's a happy little give and take or a little cycle between the chloroplast and the mitochondria, both of them giving essential ingredients to each other. All the while, what's really happening is we're taking solar energy and we're actually converting it to ATP. Life is just doing it in a very complex way using plants, autotrophs, and animals, heterotrophs, again, all for the production of ATP. All right. Let's differentiate cellular respiration, which is what we want to focus on, with breathing. They are related, but they're not the same thing. Breathing is what you and I are doing to infuse massive amounts of oxygen into our lungs. And in order to do this, we're going to use a diaphragm, right, that's just below our lungs. We're going to use our rib cage and the muscles around the rib cage, and we're going to expand them, and when we do that, it allows oxygen to move in to our lungs. Once inside, the oxygen moves into our bloodstream, and then that's how we will transport oxygen to all of our cells. You might know that the oxygen is traveling inside of red blood cells, the ones that you saw in the cell structure and function lab. Okay, so we bring in massive amounts of oxygen, we put them inside of red blood cells, and then the red blood cells take care of delivering it to all of the cells of our body. In this case, we're showing you the muscle cells of an athlete, a runner, because anytime you have an activity, that activity is going to demand energy from you. So the energy can only be extracted from the foods that we eat in the presence of oxygen. So we're going to talk about something called aerobic respiration. Aerobic implies that air is present, and the air that we're interested in, of course, is oxygen. Okay, so that's breathing. What we want to do is go inside of a muscle cell and see how that cell is taking in glucose and oxygen and using it in what we call cell respiration, again, to produce ATP. Right there. Let's get that out of the way. All right, so there's your glucose plus oxygen, giving you CO2, water, and ATP. So the goal, again, is to see how we're taking sugars, oxygen to produce ATP. That's the overarching goal. And we're going to go over this in 3 broad steps. In photosynthesis, we saw 2 broad steps, the light reactions, the Calvin cycle. Here we're going to see 3 broad steps, And you've already got flashcards for all of this, right? Okay, so here is that reaction in its entirety. Here's glucose, here's the oxygen, and notice again that it's exactly the opposite of photosynthesis, right? In photosynthesis, the arrow was pointing that way, and this was the sun providing photons to the reaction. But in cell respiration, we're going to do the backwards thing. We're going to reverse the arrow, and you're seeing it in its reversed form right here. And we're going to see that we're taking the glucose and the oxygen that was produced from photosynthesis, and we're going to break down glucose. We're in fact going to oxidize it to produce CO2, water, and again, that guy. All right. So when our cells break down glucose using oxygen to create ATP, it's not very efficient. Like, we're only extracting about 40% of the energy that's in that glucose. What happened to the 60% of that energy? Well, that energy was lost in the form of heat, right? And interestingly, we use that heat to keep our bodies warm. Our bodies are around 37 degrees Celsius. That's a good 15 degrees warmer than an average room at 22 degrees Celsius. And the reason why our bodies are warmer than the environment around us is because we're burning glucose, pulling energy out of it for our own ATP utilization, but then some of that energy is lost in the form of heat, and it's keeping our bodies warm. All right, as inefficient as our cells are at extracting energy from glucose, vehicles are even worse at extracting energy from gasoline. So the analogy I like to use is that if you put $100 worth of fuel in your engine or in your fuel tank, only about $25 moves the vehicle, and the other $75 is essentially overheating your vehicle, because again, every time you convert energy from one form to another, you always generate heat. But the amount of heat generated in an engine is so high, we have to equip the engine with a cooling system, right? Radiator, a water pump, antifreeze, right? All these liquids to keep the temperature down. And then we covered this in lecture, right? We had a good time discussing all of the activities that you could do to burn a significant amount of calories. Nothing beats running, right? Nothing. And I think you understand why nothing beats running. It involves really big muscles. The leg muscles are the biggest muscles on your body, and they burn a lot of calories. To that end, dancing fast, which you also had a good time with discussing in lecture, will also burn a lot of calories, again, because you're largely using the big leg muscles to burn through calories. So essentially anything that is showing, right, high calorie burning is usually something that involves, not surprisingly, your leg muscles. So if you're ever interested in burning a lot of calories and experiencing a little bit of a deficit in your calories, right? In other words, if you're taking in less calories than you're actually burning so that you can lose weight, the idea is you really want to do one of these exercises that I've just put an arrow to. Those are the ones that are going to get you a lot of energy burnt, and you're going to operate at a deficit and shed the weight. All right, let's remind ourselves about oxidation and reduction. Remember that oxidation is the loss of electrons or hydrogens. You might want to zoom ahead if you already know this. Oops, oxidation. And then reduction is the gain of electrons or hydrogen ions, right? I'm just circling the hydrogen to make it stand out. So in respiration, again, here's your glucose loaded with hydrogens. And then here's that same glucose now appearing as a bunch of CO2 molecules. And notice that the hydrogens are missing. So when you lose hydrogens, we can agree that this is oxidation. All right, the point of this is for you to understand that whenever you eat food loaded with carbs, lipids, proteins, you are in fact oxidizing those nutrients and pulling electrons and hydrogens out of them. Who is picking them up? Who is doing the reduction part? That's going to be oxygen. Oxygen is gaining the hydrogens that are being pulled out of glucose, and because it gains them, that's your reduction. And you might remember that you can't have oxidation without reduction, and this is why we lump together the two terms, and we simply call it redox. In the end, again, what is the ultimate goal? To produce ATP. In the process, we will create CO2, we will create water, and both of those guys are going to go back out into the world for plants to take in the CO2, for plants to take in water, right? Everything is recycled in nature. And when plants take in CO2 and water, they're going to give us back glucose and oxygen, and on and on and on. This cycle is perpetuated over time. All right, so that's oxidation and reduction, re-explained, but this time using respiration as opposed to photosynthesis. All right, we don't need these slides. Okay, I want to pause here because I want to show you how it is that we're going to produce ATP. And I wanted to show you the ATP synthase that you saw in the light reactions in plants. And I also wanted to show you the electron transport chain that you also saw in plants. We have them also. It's not in chloroplast, it's in mitochondria. So we're going to see a membrane, but it's not going to be the thylakoid membrane. It's going to be the inner membrane of the mitochondria. And that's where we are going to see the electron transport chain, the ATP synthase. We're not going to see photosystems. We do not use photons for energy. And we're going to be producing ATP. You're also going to see very briefly an example of how we can make ATP without using an ATP synthase. So this is a different enzyme. It's not ATP synthase, but it still produces ATP. We call this substrate level phosphorylation. And I tend not to make a big deal out of it. I'd rather just show you where enzymes are making ATP without the benefit of a membrane. In other words, without the benefit of an ATP synthase, right? And we call it substrate level phosphorylation. And again, you don't need to worry about this for an exam. We call it substrate level phosphorylation because there is a substrate that carries the phosphate, whereas here, the phosphate is by itself. It's inorganic. It's not attached to an organic molecule. This phosphate is organic. It's attached to an organic molecule. That's the substrate. You pull that guy out, attach it to ADP with a phosphate, and now you've got yourself your ATP, and the organic molecule changes its shape after having lost the phosphate. Again, we're going to be focusing on that guy because there's consistency with what we saw in the light reactions, and consistency is a good thing, right? We don't want to keep changing it up so that we're learning something brand new all the time. We want to keep with the things that we understand from a previous lecture. All right, so here are the 3 steps of respiration. So respiration in 3 steps. Now again, we created flashcards for this, but the first step, the first flashcard, is glycolysis. And it happens in the cytoplasmic fluid, or just in the cytoplasm. That's step 1. Here's what you should know about this step. You should know what goes in, what comes out. Again, that's in a flashcard. We're going to see it in the next slide. But here's also what you should know. This happens with or without oxygen. So what we like to say is that it is oxygen independent. Doesn't matter if it's there or not. If it's there, great. If it's not, doesn't matter. Glycolysis is going to happen anyway, irrespective of whether oxygen is present or not. Okay. I'm trying to clean this up because it looks kind of ugly. All right. Now, after you're done with glycolysis, if oxygen is present, you're going to go to the mitochondria. I'm going to be reiterating this over and over again, and I have a separate slide where I'm going to cover this again. But for now, I want you to prepare for that conversation. Remember, glycolysis is independent of oxygen. But once you're done with glycolysis, this guy right here, if there's oxygen present, this guy is going to be sent into the Krebs cycle. The Krebs cycle will operate. That's going to be step 2. And when the Krebs cycle is done, we can then go to step 3. That also requires oxygen. That's going to be where you will see the electron transport chain. You're going to see something called chemiosmosis. All of this is happening in the mitochondria. So steps 2 and 3, if oxygen is present, we'll use the mitochondria. All right, so you might be wondering, okay, well, what happens if there is no oxygen? Well, you're not going to go to the mitochondria. You're going to do something else. You're going to do something called fermentation. You and I do fermentation. We do a little bit. We still use the mitochondria because you and I can't live without oxygen. If there's no oxygen, you and I will die. And we will die because we're not producing ATP, as you will find out. No oxygen, you and I die of a lack of ATP. So when you and I are exercising and we're breathing heavy, we're taking deep breaths, like we're trying to catch our breath, that's evidence that we're not getting enough oxygen. And so that's when we're going to be doing something called fermentation. All right, so that's going to come up in a second. All right, so back to slide or step 1, flashcard number 1. You need to know what goes in, what goes out, right? We've created these flashcards already. So step 1 on the front of your flashcard will say glycolysis, which literally means the breakdown of glucose or sugars. And you should write in parentheses that this is happening in the cytoplasm. Okay? All right. And then you were supposed to have, okay, what goes in and what comes out. So what goes in is glucose. There's your glucose. What else goes in? ADP and phosphate. There they are. What else goes in? NADs, right? You need 2 sets of those. You need 2 of those. And what comes out? Well, the glucose is going to become 2 pyruvates. So I'm going to get rid of this pyruvic acid so that there isn't a couple of words now that we need to know. Let's just call it pyruvate. So there's 2 pyruvates. That's plural. And there's 2 ATPs that we make. You might recognize this reaction as being endergonic, right? You're creating a bond between phosphate and ADP. And that requires energy. That's an endergonic process. And we're taking NAD and all of a sudden it appears as NADH. You should recognize that as reduction, okay? So it's one thing to know where this is happening, cytoplasm, to know the step, glycolysis, but you should also know, okay, what are the reactants and what are the products? What goes in, what comes out, okay? And once you know that, that's memorization stuff. You can answer some questions pretty quickly, but be prepared to be able to tell me what happened there. Okay, that was an endergonic process. What happened here? That was reduction, right? And remember, reduction cannot happen without oxidation. So if NAD is picking up hydrogens and becoming NADH, where did those hydrogens have to come from? Glucose. Because remember, what do we do with the food we eat? We oxidize it, right? So glucose is being oxidized so that we can reduce NADH. Okay? Okay, that's step 1. And remember, this can happen with or without oxygen. With or without, oxygen. So we like to say that it is oxygen independent. Doesn't care. Is oxygen there or not? Doesn't matter. Glycolysis is going to happen no matter what. All right, that's step 1. All right, how is this ATP made? Let's look at a little bit of the details. All right, so first, there's the preparation phase. We eat glucose, so here's a cell. Here's glucose trying to come into our cell. And remember, glucose is an example of a monosaccharide. It is a macromolecule. And remember that cell membranes do not allow big things to go through. So we're going to need some sort of transport vesicle, and then glucose can go in. And as soon as it goes in, that glucose, now seen here in its linear form, is going to be phosphorylated. We're going to add a phosphate to it. Here it is. glucose with a phosphate on the 6th carbon. See the 6th carbon? So glucose 6-phosphate. There's some really interesting intuitive way of naming things in organic chemistry. And then we turn glucose into fructose. It still looks the same, right? It's still 6 carbons long, and the phosphate is still on the 6th carbon. Interestingly, we turn glucose to fructose. But then we add a second phosphate, and now there's a phosphate on the 6th, and now there's a phosphate on the 1st carbon. Fructose, 1,6. Diphosphate, 2 phosphates on there now. And then comes the step that gives glycolysis its name, right? Where you split that 6-carbon sugar in half, 3 carbons that way, 3 carbons this way. That's your glycolysis. Glycolysis, sugar breakdown. Okay, then comes the energy payoff phase, right? So in the previous slide, we saw the preparation phase where we invested 2 ATPs. So 2 ATPs went in. In the payoff phase, we get 2 ATPs from this one step, and then we get 2 more ATPs from this second step. So 2 ATPs were invested, 4 ATPs were returned. That's a net gain of 2 ATPs. And that's why you see those 2 ATPs there. All right? So 2 ATPs invested, 4 come out. And then here's the part that does the reduction step. Okay, how are we making ATP here? There is no membrane. Remember, glycolysis is happening in the cytoplasm. So how did we make ATP? Well, we made ATP by pulling the ATP out of an organic molecule that had phosphates, right? We pulled that ATP out. That should look familiar. We saw it right here. There's an organic molecule with a phosphate. We pull it off and we make ATP. That's called substrate level phosphorylation. So that's another way of making ATP. But I don't need you to worry about that. I need you to worry about ATP synthase. We're going to get to that guy. That's the guy that gives us the massive amount of ATP that you and I are largely operating with. This ATP that's made with this enzyme, it's very little ATP, all right? Okay, so we just saw glycolysis. What's happening in this slide? Well, glycolysis produced this guy. Again, we're calling it pyruvate. Let's clean that up. Pyruvate. Okay, so that came from glycolysis. We turn glucose into pyruvate using glycolysis. And remember, this happened in the cytoplasm. As soon as sugar comes into a cell, we're breaking it down. But it turns out that to go to the second step, step 2, the Krebs cycle, to go to step 2, you need to convert pyruvate into something that the Krebs cycle recognizes. It does not recognize pyruvate. So you have to convert pyruvate, here called pyruvic acid, but again, let's just go with pyruvate. It has to be converted into acetyl-CoA. So here's what I want you to know for exam number 2. If you should be asked, how do we connect glycolysis to the Krebs cycle, your answer should be, in the multiple choice, that we need to convert pyruvate to acetyl-CoA. You can bank on one question asking you that. So know that. Now, while I have your attention on this, notice that NAD is becoming NADH. That's being reduced, right? So who's being oxidized? What's left of sugar. Also notice that we're breaking off one CO2. 1/3 of the CO2 you exhale comes from there, from this step. And then notice that there's a coenzyme A that's being added. That's vitamin B5. We're going to add that in here. So acetyl-CoA is actually acetyl vitamin B5. That CoA, that coenzyme A, is actually B5. The vitamin Bs are very important in energy reactions in our cells. Okay, again, what's the big takeaway? If you want to connect glycolysis to the Krebs cycle, you need to convert pyruvate to acetyl-CoA. That's the takeaway. All right, let's go now to the Krebs cycle. Now remember, we're in the Krebs cycle because oxygen is present, right? Otherwise, we can't go to the Krebs cycle. This is the Krebs cycle. This is step 2. And again, on your flashcard, you should have, this is Krebs, and it is happening in the matrix of the mitochondria. All right. What goes in, what goes out? All right, follow your arrows. First thing going in is that important acetyl-CoA. What else is going in? NAD is going in. ADP and phosphate are going in. What else is going in? FAD is going in. All right, what's coming out? Well, what happened to that acetyl-CoA? It was turned into CO2. You see it here. What happened to NAD+? It became NAD with an H, so it was reduced. That means you're oxidizing that guy. We're oxidizing the food we eat in a variety of steps. What happened to ADP and phosphate? I think you can predict that happens to become ATP. And then what happened to FAD? Well, that became FADH with a 2. That's what comes out. All right, that's pretty much all you need to know. You need to know what step 2 is called, Krebs cycle. You need to know where it happens, the matrix of the mitochondria. You need to know what went in. You need to know what came out. And finally, you need to know that this requires oxygen. Otherwise, you can't go into the mitochondria. All right, this leaves the last step. So for the last step, we need to take what comes out of the Krebs cycle to feed the third and final step. And what we're going to feed it is this guy, NADH and FADH2. So let's go to the last step, but I'm going to draw the last step. And then I'm going to show you the actual diagrams. And then I'll include a diagram of the last step on the exam. It won't be the exact same diagram, but it'll look very similar. In fact, not similar. It'll be like that diagram, but maybe I won't include labels. So you're going to get visuals to help jog your memory. You won't be relying on memorization. All right, let's go to the third and final step. This is going to be called oxidative phosphorylation. And we're going to feed it NADH and FADH2, right? There's going to be some other things we'll feed it. But let's go there. Oh, by the way, FADH2, if you were wondering, is vitamin B2. All right, so we've seen all kinds of vitamins. Turns out NADH is vitamin B3. FADH2 is vitamin B2. And then earlier in the previous slide, right, you saw acetyl-CoA, and that's vitamin B5. Okay, let's go to that last step. Let's just do a blank screen, and we'll just sketch the whole thing. All right, so first, where is this happening? Okay, here's the mitochondria. And remember, the mitochondria has an inner membrane. That's that wiggly one. And then it's got an outer membrane. So that's the outer membrane. And then there's your inner membrane. And then in the liquid inside is the matrix. This is where we just came from. This is where the Krebs cycle is happening. All right, so this is your mitochondria. I'm just going to write mito here. What's outside the mitochondria? That's the cytoplasm. That's where you were doing glycolysis. The only reason why we're in the mitochondria is because presumably oxygen was present. So here's what I want to do. I want to take this part of the membrane. Maybe I'll make it red so that it pops. I want to take that inner membrane, and that's what I want to magnify, right? So we're in the inner membrane. Like, we can just show you the whole thing. We're going to focus on that inner membrane. And maybe I'll focus on that part of the membrane. So if you're following along, here's what I've got going on. The top line, I want that to represent the outer membrane. And in red, let's say, I'm going to make this one broader or thicker. This is the inner membrane. So I'm trying to keep it color-coded so you can follow along. All right. There's going to be an electron transport chain, and there's going to be a ATP synthase. So we can label those. And then these guys right here represent the electron transport chain, ETC. All right, this should look familiar. This looks like the light reactions, like the thylakoid membrane, except that we don't have photosystems. So it ought to be simpler, I would think. All right, what's going on in the space above the inner membrane and the space below? What do we have there? Okay, well, in the space below, we're in the matrix, right? So all this is the matrix. And we just got through doing the Krebs cycle. That was step number 2. That was happening there, right? It was happening here in the matrix. Okay, what's above that? What's above the inner membrane? Okay, we're going to call this space the intermembrane space. It's literally saying it's the space between two membranes. So there's like really nothing creative about that name. All right, I think we're ready. All right, so the Krebs cycle gave us a couple of important products. It gave us NADH and it gave us FADH2. Both of those things came out of the Krebs cycle. Those guys are carrying electrons, and we know because all of a sudden they have hydrogens that they didn't have before. So the electrons are going to be donated there on the electron transport chain. And when they donate their electrons, the electrons are just going to move through redox. And as the electrons are moving through redox, hydrogen ions are going to be moved or translocated from the matrix into the intermembrane space. So now we've got a whole bunch of hydrogens in here. Now it's acidic in the intermembrane space. And the idea is that those hydrogens will then fall back down to the matrix from high concentration to low. And they're using the ATP synthase as a facilitated transporter. That's what it's doing. Because remember, hydrogens have a charge. They cannot get through the membrane, any membrane, by themselves, so they need help. And in this case, the ATP synthase isn't just an enzyme making ATP. It's also a facilitated transporter. It's allowing hydrogens to go from high concentration to low, from the intermembrane space back down to the matrix. As soon as those hydrogens get down into the matrix, guess what happens to them again? Boom, they get thrown back up, they come back down, and they just keep doing that. As long as we keep putting electrons into the electron transport chain, hydrogens keep moving across the inner membrane. They fall back down through the ATP synthase, and they keep going up, back into the intermembrane space. Okay, while all this is happening, right, ADP and phosphate are being linked to create the oh so important ATP. So that's the purpose of the hydrogens going from high concentration to low. It's all providing energy so that you and I can make that ATP. But again, electrons have to be continuously going into that electron transport chain. And where did those electrons come from? They came from the foods you ate. We oxidized those foods. We pulled out the electrons. Did you see the electrons being pulled out of the food? No. Instead, you saw hydrogens being pulled off of molecules. But that's the evidence of the electrons also coming off. All right, so to complete this diagram, we need to remove this electron from the final electron transporter. That electron has to come out. And the person who's going to pick it up, I said person, but you know what I mean. I mean, who's going to pick it up? It's going to be oxygen. And when oxygen picks up the electron, it'll pick up two acids, and we will make H2O. So here's what I need you to know. I need you to know that the electron transport chain is actually working backwards from what you saw in the thylakoid membrane. Go back and review it. Or you might remember who provided electrons in the thylakoid membrane. It was water. And what happened to water when it gave up its electrons? It became all this. And who picked up the electrons at the very end of the light reactions? It was the cousin of NADH. It was a guy called NADPH. But here it's all backwards. NADH drops off the electrons there, and the electron is picked up by oxygen to regenerate water. See, we're doing everything the opposite of what plants did. Okay, you might want to go back and review this. Yeah. But remember, this is called step 3. It's called oxidative phosphorylation. That's step 3. And it requires an electron transport chain, seen there. It requires an ATP synthase, seen there. And hydrogens need to go from high concentration to low so that we can generate ATP. That has a name, and you may not like it, but that name showing hydrogens going from high concentration to low is called chemiosmosis. And you also saw that in the light reactions. All right, so if you wanted to create a flashcard, you're definitely going to need this part. You should know that it happens in the inner membrane of the mitochondria, right? We've just been drawing it right here. And you should know what went in and what came out. What two things went in? NADH went in, FADH2 went in. What else went in? Oxygen went in. And then what came out? Oh, by the way, ADP and phosphate also went in. And then what came out? Well, what came out was water, right? And ATP. We also created NAD+. After NADH donates its hydrogens, it becomes NAD+. After FADH2 donates its hydrogens, it becomes FAD. So create your own flashcard there. All right, that's a complex diagram, right? You got to give this time. You got to replicate it over and over again. But I don't want you to think about memorizing stuff. Some things you'll memorize for sure. The name, oxidative phosphorylation, that it happens in the inner membrane. You'll remember that no matter what. But what if I give you the diagram? Right? I can give you the diagram, some parts labeled, but this is your electron transport chain right here. Here's your ATP synthase over there. See the NADH donating electrons and becoming NAD+, FADH2 donating electrons becoming FAD. Here's the oxygen picking up electrons becoming water. Here's ADP becoming ATP, as long as you've got hydrogens going through. Here's your labeling. And then here's the step, oxidative phosphorylation. Here's where it's happening, right? Inner mitochondrial membrane. Here's your outer membrane over at the top. Replicate these diagrams. It's a good way to get them down. This is what the ATP synthase looks like up close. The hydrogens represented by this little sphere. They actually bind somewhere on the ATP synthase, and then there's a motor protein that spins. It's really incredible. And when it spins, it generates ATP. It's super, super interesting. All right, that's like a whole lecture can be done on just ATP synthase, and we can do that like in a bio majors course or a molecular bio course. This is a much more simplified image of the oxidative phosphorylation step, step 3. It's got the electron transport chain, it's got ATP synthase. And things are just a lot cleaner, you know, a little more cartoonish, right? It doesn't have to be like a complicated diagram like that you're drawing, right? But when you draw it, eventually, it'll look pretty complex. But this is a nice simple one. You know, you can look at this one. It's a lot more gentle on the eyes, I would say. All right, so we've done now the 3 steps of aerobic respiration, culminating in this third and final step called oxidative phosphorylation that's happening in the inner membrane of the mitochondria. In the next video, I'm going to show you fermentation. All right, so let's end it here. Whoa.